Here we are, ladies and gentlemen, the ultimate video in our series on learning about the human body and all of its icky parts. We're going to be talking about genetics and inheritance. So not a whole lot of like body part type stuff. It's the whole genetic part, that little instant of fusion between egg and sperm where conception takes place. We're going to talk about all the various ramifications for that and stuff like that. So without further ado, let's review our genetics from our freshman year. Okay. So I want to throw a few terms at you just because I can. So a couple of terms to talk about. Differentiation, I mentioned it before. It's the creation of different types of cells during development, and there's selective changes in genetic activity. This gene gets turned on, this one gets turned off, and vice versa. Okay. We've got inheritance, also known as heredity. That's the transfer of genetic information from generation to generation, from parents to offspring, that sort of thing. And genetics is the study of these mechanisms responsible for moving this information along. And so that's basically what we're going to be doing. Where we find all this genetic information are chromosomes, which are structures that contain DNA and proteins. Within DNA, you have sequences of base pairs of DNA known as genes, and each gene carries specific information needed to create a specific polypeptide, a specific protein that is then going to go on to do a certain thing. And that's basically all life is. I'm sitting here talking to you right now. I'm just I'm a result of selectively activated genes creating proteins. I mean, that's it. It's a sort of elegant way to describe a lot of the stuff that has going on. Or particularly Nietzschean. I guess it depends on where your sensibilities lie. When we're talking about genetics and genetic description and stuff like that, we want to talk about genotype, which is the genetic expression, the actual chromosomes and their genes themselves. What does the DNA say? The phenotype is the message, the physical manifestation, the physical description of the genes. Those are the characteristics, the physical characteristics, the behavioral characteristics, anatomical characteristics, stuff like that. Okay. Your phenotype is going to be determined by your genotype, but also your environment. That's something we're going to talk about later, kind of towards the end. So each one of our cells has 23 pairs of chromosomes. Okay. At amphimixis, each pair is contributed. One member of each pair comes from mama, the ovum, and one comes from the sperm. 22 of those 23 are known as autosomes, which are consisting of homologous chromosomes. So they're pairs of chromosomes that are basically the same. They have the same structure, they carry the same genes, and stuff like that. Most of the genes are going to affect somatic characteristics, things that we can't control. The remaining pair, that 23rd pair, are the sex chromosomes. That's when it's going to determine if we are genetically male versus female. Pause. That doesn't include, that doesn't necessarily determine, are you a man or a woman? Those are social constructs. So we're talking about male or female. There's your sort of soapbox moment. The more you know, star, if you've seen those memes. Okay, so these are your chromosomes. Well, not yours specifically, but these are human chromosomes, okay? You've got the numbered ones. Those are the autosomal chromosomes. We've got 22 of them. As you notice, they get successively smaller. And then the last one, it's unlabeled. It's not numbered. It's the X and the Y. So that means genetically, this individual is male because they have the Y. Females will genetically have 2 X's. When you're looking at this, a lot of medical professionals will make a karyotype of an individual to look for structural deformities or chromosomal deformities. So for example, if you are trisomy 21, tri meaning 3, so there's 3 copies of 21. That is Down syndrome. That is a chromosomal disorder because you have the wrong number of chromosomes. If you have one less of some chromosomes, so you only have 1 copy, that's another one. And so basically you can look at this visually to see if there's any issues. Okay. The locusts, not the locust, the locusts usually come in plagues and eat everything. A locus is a specific position on the chromosome. Typically, it's where a gene is. It's its location, like its address. Genes can have alleles, which are different versions, right? A and B, big A, little a, that sort of thing, right? How we pair up these alleles is what determines the precise effect of the gene on a particular phenotype. We're going to get into that in a little bit, okay? We can have 2 alleles that are the same, that is homozygous. You can be homozygous dominant, that means they're both big, or they can be homozygous recessive. Heterozygous, hetero meaning different, zygous meaning like zygote, like different arms, different chromosomes, okay? Homo is the same. If you are heterosexual, you are sexually attracted to somebody different than your sex. If you are homosexual, you are attracted to somebody of the same sex as you, okay? So homozygous, the same alleles, okay? Heterozygous, 2 different ones. We typically use capitals and lower cases, okay? So heterozygous would be capital, lowercase. Different patterns of inheritance. We can have simple inheritance. Phenotype is determined by the interaction of just a single pair of alleles. Now within that, you can still have a lot of stuff going on. For example, you can have strict dominance. Strict dominance is what we think of, okay? So if something is dominant, that allele is always expressed. It's going to cover the recessive allele. The only way you can have the recessive allele is if you don't have any dominant alleles, right? So homozygous recessive is the only way to get a recessive phenotype. By the way, if I'm moving very fast on this stuff and you need a little bit of a slower refresher, Amoeba Sisters has a lot of really good stuff on how all this works. Okay, they could break it down for you a little bit better, and they look cuter. The animations are better. So we've got an example that we're going to work. So albinism is a recessive allele to normal skin pigmentation. So albinism is no pigmentation. So no skin pigment, no eye pigment, no hair pigment. Okay. If 2 heterozygotes have children, what is the chance that a child will have normal skin pigment? And what is the chance that the child will be albino? And so we're going to use a Punnett square to do that, right? And so we know they're heterozygote. So I'm just going to say, let's just use A, okay? Whatever, I don't know. So big A is dominant. That is having skin pigment. Little a is recessive. That means you are albino, okay? 2 heterozygotes have children. So we know that heterozygotes are different, right? So I'm going to put one parent's alleles on the top, one parent's gametes on the top, one parent's gametes on the side. Doesn't matter where the mom goes, where the dad goes. It doesn't matter. We're going to fill the boxes in, right? So I want to know what the chance is that I'm going to have a child with normal skin pigment. So that means a child with at least 1 dominant allele. So I've got 1 here. I'm going to fill that in. I'm going to fill that in. I'm going to fill that in. Okay. So the first question, what is the chance that a child will have a normal skin pigment? If I look at this, I've got 1, 2, 3 out of 4 possible scenarios. That means I'm going to have a 75% chance that my child will be normal. What's the chance that the child will be albino? Well, I've got 1 out of those 4. So that means I've got a 25% chance that my child will be an albino. Pretty easy, right? The next one, we've got codominance. Codominance, where heterozygous individuals exhibit both phenotypes. We have equal expression of the phenotypes. It's not one masks the other, it's both of them show up at the same time. A pretty classic example in humans is blood typing. So so here's my blood type. I've got a man with AB is going to marry a woman with type B. We know that mom's mom is type O. We want to list the genotype and phenotype frequency. So we want to list off the possibilities, basically, is what this is saying. Now, the reason why we put mom in there is because we need to know what type of grandma. The reason why we put grandma in there is we want to know what type of bee mom is. Okay, so if we look down here with B, you can either be IB or just big B. I typically use AB and little I, but it doesn't matter. I can be IB or IB little I, and I'm still type B. Okay. Because right now we've got a little bit of strict dominance. Okay. If I've got AB, here's the codominance. Both are expressed equally. So I've got an equal number of A's and equal numbers of B's. We talked about this when we did the blood. Okay. So I've got dad, who is AB, and mom is O. That means if mom is O, we can say, if grandma is O, that means mom is, we can say is big I, B, little I. Okay, so I'm going to use it as B, little I. And so I'm going to figure out what my possibilities are. So I have an AB, I have a B, I have an A, and I have a B. So those are my phenotypes, right? My phenotype frequency is right there, and you would list that off. My genotype frequencies, well, in this particular case, my genotype frequencies and my, well, no, here, let me back up. So I misspoke. Oh dear me. Okay, so my genotype frequencies are what you see here. My phenotype frequencies, because if I look at this, those are the same phenotypes. They're both blood type B. So my phenotypes are going to be AB, A, and B. No O's, because both parents have to have an I allele in order for them to be a type O parent, a type O kid. Okay. What else? Let's move right along. How about incomplete dominance? So heterozygous alleles have intermediate phenotypes. This would be like a blending of colors and flowers, for example. So in humans, we've got sickle cell anemia, okay? We've talked about that when we did the blood. It's a genetic disorder. So hemoglobin gets malformed, which caused the blood cell to become sickle-shaped, not that sort of like lifesaver donut shape. Let's see, what are the possible genotypes and phenotypes of offspring of sickle cell carrier parents? So something to keep in mind, if you see the word carrier, that means they are heterozygotes. They have one of the alleles. They are carrying the allele, but they themselves do not have that particular phenotype. They can give it to their children. Okay, so that means, what's a good letter? I'm going to do B's. I'm going to say big B is regular, and I'm going to say little b is sickle shape. They are carrier parents. That means they are both heterozygotes. What are the possible genotypes and phenotypes of my babies? So I'm going to fill in the boxes. And look at that. So possible genotypes is what you see here, okay? Possible phenotypes are what those expressions are. So that one, and that one are the same phenotype, regular blood cells, okay? So 3 regs and 1 sickle cell. Those are my phenotypes, but my genotypes, it's 1 homozygous dominant, 2 heterozygotes, and a homozygous recessive. A 1 to 2 to 1. What's some other ones? Polygenic inheritance. There's multiple different genes that are going to determine the same trait. You can't use a Punnett square because the interactions are too complicated. You've got multiple genes. Lots of risks for hypertension, coronary artery disease, and stuff like that are polygenetic. There's a lot of different markers that will determine this. So it's very complicated. Are you going to have coronary artery disease? You can't just pinpoint it. Like hitchhiker's thumb, that's just one gene. Either you have it or you don't. Same thing with attached earlobes. Here's an example. Skin color is another one like that. In this particular case, it has to do with the number of genes that are activated. The more genes that are activated, the darker your skin color. The fewer genes that are activated, the lighter your skin color. Basically is what it boils down to. Another type of polygenetic inheritance is known as epistasis. Epistasis is when you have one gene that acts as a suppressor. So let's say you've got one gene that, hair color for various organisms is an example of one. One gene codes for the amount of pigment put into the individual hairs. The other gene acts like a gatekeeper. That's going to allow pigment in the first place, right? And so if that door is closed, that gene is going to suppress the implantation of that pigment. So it doesn't matter how much pigment you're supposed to have, that door's shut. Nothing's coming in. You can also have dominant alleles with different genes will actually interact to produce different phenotypes than the other genes. So here's an epistasis example. These are Labrador retrievers. So if you look, the B is how much color. So homozygous dominant is the most color. Homozygous recessive is the least amount of color. So all your black labs are your dominant phenotypes. Your chocolate labs are your recessive phenotype. In order to have that color put into the hairs, you have to have a dominant phenotype for the E. That's the gatekeeper. So that's why if you look, all of our yellow labs are homozygous recessive for that gatekeeper gene. It works similarly in humans with red hair. There's two pigments, eumelanin and pheomelanin. Eumelanin is your brown and black. The more eumelanin you have, the darker your hair color is. So black hair has a lot of eumelanin. Pheomelanin is primarily red. And so if you have a certain gene, that's going to convert all of your pheomelanin to eumelanin, and so you're going to get your blondes, which don't have a lot of melanin, your browns, which have some, and your black hair, which is a lot. If you change how you express that gene, you're not going to produce any eumelanin. It's all going to be pheomelanin, so your hair is going to be white or red. So like your strawberry blondes are going to be sort of a blending of that also. So that's another example of epistasis, and that's all polygenic. We also have sex-linked chromosomes. Most of the time we're going to talk about X-linked. There are some very rare cases where it can be Y-linked, but not usually. So your X chromosome, it kind of looks like an X, that's why it's called that. It's significantly bigger than the Y chromosome and has more genes on it than the Y chromosome does. It's carried by all of the oocytes. So basically, mama is going to supply an X. Okay, your Y chromosome, it has genetic information specifically coding for genetically male. There's a whole lot of stuff missing. So if you are a Y, you are genetically male. That comes from the dad. Now, dad can also give an X, so two X's make you genetically female, okay? So it's dad that determines the genetic sex of the baby. So X-linked genes are genes that are found on the X chromosomes. They're not going to be found on the Y chromosomes. They can affect some structures, somatic structures. Somatic structures are not sex-related structures. So basically, the structures that don't have to do with the actual formation of the eggs in the gonads. Inheritance doesn't follow patterns of alleles and autosomal characteristics because you've got different percentages based on male versus female. For example, hemophilia is located on the X chromosome. It's a recessive disorder, and so it's similar to other recessive disorders. However, what we want to do is we want to see why is it less likely for a woman to develop hemophilia than a man? And we're going to have to keep track of what sex the children are. So let's see. A man is normal for blood clotting, and a woman is a carrier. So a man is X big N Y. Mama is a carrier, so that means she's big N, little n. Let's figure out the offspring. All right, so we've got one daughter who is normal blood clotting, not even a carrier. We've got a son, potentially, who is normal. We've got a carrier daughter and a son who has it. And so if we're looking at this, there is a 0% chance that any of their daughters are going to have hemophilia. There is a 50% chance that their sons will. And so that gets into why is it less likely for a woman to develop something like this? I'll give you a second to think about it before I give you the answer. Give up? It's because in order for a woman to have hemophilia or any X-linked recessive disorder, both have to be recessive. Just like regular simple or just like regular strict dominance. In males, if you have the gene, you have the disease. There is no other gene to balance that, to offset that. If that gene is there, that's what's being expressed. Too bad. That's why in royal families in the 17 and 1800s, you saw hemophilia striking almost exclusively in the males, not the females. There were a few instances, and there's a really interesting case study. If somebody reminds me, I could put it up for y'all to look at. Basically looking at how Queen Victoria and her consort could have led to the downfall of the Russian aristocracy and the rise of communism. So it's an interesting read and it's an interesting activity. I'll try to remember to put it up. It's pretty good. So sources of individual variation. Chromosomes are randomly distributed during meiosis, so that's one thing. Each gamete produced has a unique combination of genetic material of maternal and paternal chromosomes. So it's a crapshoot, basically, as far as who gets what chromosomes, which egg and which sperm comes together. You can also have something called genetic recombination, which is various changes will occur to the chromosomes themselves during meiosis. And so you're going to have chromosomes that are different from the parent. They all came from the same source material, but they are genetically different. And so that's going to also increase variation. And so here's an example. This is crossing over, basically. Two chromosomes are laying next to each other. They're going to stick their legs out, and they're going to cross over. And that's going to cause this arm right here and this arm right here to switch places. And so when we move back, a little bit of this chromosome is right here, and a little bit of this chromosome is right here. Same genetic source, but these are not chromosomes that were produced by the parent prior to this. This happened during meiosis. Here, you can also have chromosomal abnormalities, which would show up on that karyotype that we mentioned earlier. Damaged, broken, missing, or extra copies of chromosomes. These are present in 10% of zygotes. Most of the time, they're not going to survive. So we're down to 0.5% of newborns. And so there's a variety of serious clinical conditions, congenital birth defects. Down syndrome, for example, is a very common example of this. What else? What else? We're still talking about genetic recombination. So genetic imprinting is a phenomenon. Your DNA doesn't change, but you have reversible modifications to the DNA and its proteins, not the actual sequence of the DNA itself. This is going to determine whether a gene is expressed or not. So we haven't changed how the actual structure, we're changing how we read it. We're changing the instructions of the book. This gets us into something called epigenetics. That's the study of inherited traits that are not due to the actual DNA sequences. And it's specifically by activating or inactivating specific genes. It's how you read the instructions. Let me give you an example. This is my favorite one. I'm going to give you 2 sentences, and I want you to think about what the differences are. So here's the first one. So take a look at those 2 sentences and tell me what the difference is between the two as far as the meaning is concerned. Because when I read those, the first one, it says, Grandpa is coming for dinner. We are going to eat him because he's tasty and he's old. The other one, we're saying, Grandpa's asleep on the chair in the living room. He needs to come eat. We changed how we read the sentence, and so we get a very different meaning from our instructions. And that's basically epigenetics. Please, somebody give me my Nobel Prize now. I'm still waiting for it. Nobody's given it to me yet. So if y'all know anybody in the Nobel Prize community who can give that to me, that would be stellar. You can have mutations. Mutations are changes in the nucleotide sequence of an allele. They can be spontaneous, which are just random errors in DNA replication, because replication's happening literally trillions of times in our lifetime. It's common. Most cases of error are going to be repaired by enzymes in a very elegant process that you can read about later. Errors that go undetected can change the phenotype. A lot of them won't because of built-in redundancies in how we read the code. However, if it goes far enough and it happens in your gametes, in your gonads, it can produce gametes with abnormal alleles, but beneficial alleles as well. Let's see, we are almost done. Penetrance. Penetrance is the percentage of individuals with a particular genotype that show the expected genotype. So if they're big A, little a, we expect them to be big A, little a the dominant phenotype, for example. Expressivity is the extent to which a particular allele is expressed when it's present. So maybe it's a highly expressive allele, or it's not a very expressive allele. It doesn't show up that much. It gets masked by other things going on. Remember that whole polygenic thing? Chemicals that can result in abnormal development are known as teratogens. Thalidomide, which was a tranquilizer given to mothers in Britain in the 80s as a classic example, it led to gross, both large and disgusting, structural abnormalities in babies that were born during that time. Missing arms, missing legs, multiple fingers, stuff like that. Google it. It's a really interesting read. This is the human genome. Back when I was in college, before you were born, this was a big deal. They wanted to actually see what the sequence of DNA was. And it started out, there's this huge text file. It's just a string of random ATGs and Cs. And then they went through and they were like, okay, well, what about the proteome and all of this other stuff? What do the actual sequence mean and stuff like that? And so basically what we've done is we've looked at, these are all genetic disorders that we know have a specific genetic cause. And we know exactly on what chromosome and what locus that particular disease comes from. And a lot of these diseases, where right now the cutting edge is gene therapy, actually finding ways to physically alter one's genes to insert genetic information so you do not manifest these diseases, these cancers, color blindness, hemophilia, Marfan, which some of y'all researched way back. at the beginning of the school year. Lots of really good research going into that. So if this is sort of, tickling your fancy, going into that sort of molecular research, there's a lot of really cutting edge, but there's a lot of ethical considerations too. Where do we draw the line at changing the gene that codes for Huntington's, which is an incredibly devastating disease that strikes later in life, and so it transmits sometimes more frequently than other. That's a autosomal dominant disorder. Where do we draw the line from changing those genes to maybe making designer babies? What if I want my child to be smarter than he or she would normally be, faster, stronger? That's a really sticky situation that you can start a lot of really interesting ethical conversations on, if that's the sort of thing you like to do, if you like to stir that pot. That's basically genetics. We went through pretty darn quickly. I would recommend go back through and rewatch this if you need to. See me during office hours if you want to work any specific examples or anything like that, to kind of go through some of this stuff. Amoeba Sisters has a lot of basic stuff for going over a lot of the stuff at the beginning of this video clip. This was just a super quick rundown of everything. If you have any questions, send me an email, send me a reminder, hit me up in the office hours. Hey,